objective 2, which is explaining the ionic basis of an action potential, and then describing the gating mechanisms for our voltage-gated sodium and potassium channels. Now we're going to see this illustration a lot. It's very important. So I would sort of flag it. Again, something you should be able to draw or reproduce on an exam to help you. And we can see some reference points here that we know already. The equilibrium potential of sodium, which is what in terms of a value of the membrane potential? What's sodium's equilibrium potential? It's positive 60, right? So positive 60, way up here, very positive. And again, the membrane potential, I keep going back to these sort of points that we mentioned in Unit 1, but the membrane potential is describing the intracellular environment with respect to the extracellular environment. So when we say we have a positive equilibrium potential up here, this means that we have a positive environment inside the cell when sodium is at equilibrium. Okay, really important for you to understand what these terms mean. And then we can also see some other important reference points. Potassium's equilibrium potential, very, very negative, negative 94 millivolts. And then somewhere in between here, but again, more reflecting sodium, is the resting membrane potential. Now we can see several different events around the resting membrane potential. There are depolarizing events. There are repolarizing events. And then there's the resting membrane potential. And there are hyperpolarizing events. Very important to associate these terms with the direction of change of the membrane potential. So positive changes of the membrane potential are depolarizing or excitable events. Negative changes of the membrane potential are repolarizing events. And then changes that exceed the resting membrane potential in terms of being more negative than the resting membrane potential are hyperpolarizing events. Okay, hyperpolarizing events. Now, we also need to loosely describe how this movement is taking place. All right, sometimes students often lose sight of the big picture of why is the membrane potential changing? What's causing the fluctuation of this membrane potential to begin with? And we need to understand what the membrane potential is in order to answer that question, okay? It's that separation of charge. And so therefore, the movement of particles. Now, these are charged particles, so they require channels, right? Ion channels. And so basically, when we talk about the fluctuations of the membrane potential, all we're saying is that the membrane is becoming more or less permeable to some of the ions in question. If it becomes more permeable to potassium, it's going to start dropping. If it becomes more permeable to sodium, it's going to start rising. And so we have to have an understanding of what's causing those fluctuations, right? A depolarizing event, we're approaching sodium's equilibrium potential, simply means to us that the membrane is now more leaky, more permeable to sodium. And so the membrane potential will start to look more like sodium's equilibrium potential. Okay, so that's all these fluctuations are, right? Fluctuations of the membrane potential as a result of changes in ion permeability, specifically the permeabilities of sodium and potassium. Let's speak about all the channels that we've encountered, and let's talk about the voltage-gated ones. We haven't spoken about voltage-gated channels so far. We've spoken about leak channels, right? These are channels that are always open. They're throughout the entire length of the neuron. And they're most significant in establishing the resting membrane potential. We just drew that out and saw how those potassium channels and that sodium channel was critical in establishing the resting membrane potential. So those are our leak channels. Then there are ligand-gated channels. And to describe what a ligand is a molecule that binds to some type of channel or receptor and causes a conformational change. So a ligand-gated channel will only open if something binds to it, right? Some molecule or some sensor interacts with this channel. We'll talk about these when we look at the postsynaptic communication, because these are actually on the dendrites and the cell body and are very important in receiving the stimulus from that postsynaptic neuron. So let's say that again. The ligand-gated channels are going to be found on the soma or the dendrites of a neuron because they are on the postsynaptic side. Okay, so think about our two neurons communicating. the axon terminal of that first neuron, our presynaptic neuron, communicates with the soma of our postsynaptic neuron. And on the cell body of that second neuron, we have lots of these ligand-gated channels because we have molecules that are crossing the synapse to open those channels. Voltage-gated channels, very, 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 very important. If you're going to highlight, underline, underscore, underline any of the channels, it's going to be the voltage-gated channels. They can be a little bit tricky to understand. We're going to talk about the different configurations and the different states of these voltage-gated channels. But the bottom line here is that these open or close in response to fluctuations of the membrane potential. So let's say we have channels, voltage-gated sodium channels, when the membrane potential goes above a certain threshold, which we know to be -55, these channels will open up. So they are sensitive to the voltage or sensitive to the membrane potential. There are two types, sodium and potassium. They're found most throughout the length of the axon, especially at the axon hillock, and we talked about why. That's a very important area of adding our various graded potentials and deciding whether or not we're going to fire an action potential. And they are essential in establishing, firing, propagating an action potential. Okay, so whereas our leak channels are more contributing to the resting membrane potential, these are important in action potentials. And then finally, calcium channels are going to be on the axon terminal. These are responsible in the release of various neurotransmitters. So basically that final communication where one neuron speaks to another neuron. Okay, so it's important for us to distinguish these types of channels. Okay, so let's speak about ion gating along the axon. Now, as you mentioned before, membrane potential changes are controlled by how permeable the membrane is to that ion. And that is nothing more, right? Don't complicate that in your mind. That's nothing more than how many channels are on this neuron, right? So potassium has two types of channels. Not gated or always open. These are called potassium leak channels. We encountered these. We talked about these. We drew these out earlier. These are those channels that are 25 times more present than sodium. And they're basically leaking leaking along the concentration gradient of potassium, working in coordinates with that sodium-potassium pump. Whereas the voltage-gated potassium channels, these are not always open, okay? They are only open at a specific voltage. We'll talk about what that number is. But when the membrane potential gets to a specific threshold, then these voltage-gated channels will either open or close. We'll talk more specifically about their different configurations. Sodium has only voltage-gated channels that are closed at rest, and the membrane is less permeable to sodium at rest, okay? So basically, our voltage-gated channels for sodium are closed at rest, right? We'll talk about how that is. We have two different gates on our sodium channels, and we'll look at the different states of those gates.